Okay, biopsychology revision. You're probably watching this when you're panicking just before the exam, so let's get this shiz done. Right, so start with the nervous system. So you need to know about the central nervous system, the CNS, and this is the brain and the spinal cord. Obviously the brain is split into two hemispheres and the cerebral cortex. We'll go into more details about that in a moment. And then you've got the spinal cord, which obviously runs throughout the body with the nerves and reflex actions to your muscles. You've then got the peripheral nervous system, and this sends messages to and from the central nervous system via neurons. And again, we'll find out a bit more about those in a moment. But that's got two branches. The peripheral nervous system's got two branches. You've got the autonomic nervous system, and that's almost seen as your involuntary, your unconscious, vital for survival tasks. So your breathing, your heart rate, your digestion, and this is known as affecting your smooth muscle reflexes or actions. This is further split, and again we'll go into more detail about this in a moment, into the sympathetic and the parasympathetic sections of the autonomic nervous system. We'll come back to that in a moment. The other side of the peripheral nervous system is the somatic nervous system, and this is where you get your sensory and muscle information. So this is your senses: touch, taste, smell, etc. And then your muscle information. This is your skeletal muscles. This is your movement, and this is generally voluntary. You want to go and pick up the glass, you pick up the glass. Enough terminology for you yet? Just wait, there's more coming. The next thing you need to be aware of is your endocrine system. So the nervous system is obviously to do with your nerves and neurons, as we say. The endocrine system is more to do with hormones. So hormones are chemicals secreted into the blood, and they tend to affect cells and organs throughout the body. You need to know about a few different glands. So the pituitary gland is seen as almost the master gland because it affects all the others. So you've got your thyroid, your adrenal gland, pancreas, ovaries, testes, etc. And again, there'll be more about those coming up. Something you need to be aware of, which is almost the interaction between the endocrine system and the autonomic nervous system, is the fight or flight response. And so there I've got a picture of some birds which fly fighting. Do you see what I done? So the fight or flight response, you need to be aware of this. We need to go into a bit more detail about the peripheral nervous system and the autonomic branch of the peripheral nervous system. So you've got the sympathetic nervous system, which is part of the autonomic nervous system, and that increases your heart rate, it increases your breathing rate, increases your sweat. That's obviously meant to be sweat, spelling mistake. Dilates your pupils, inhibits your digestion, inhibits saliva production, and it's what Alex Ferguson would call your squeaky bum time, contracts your rectum. And so this is the one that gets you ready, gets you hyped up, gets you pumped. You've then got the parasympathetic nervous system, and this is the one that calms you back down, basically. So it decreases your heart rate, decreases your breathing, constricts your pupils, stimulates your digestion and your saliva. It's the kind of your resting state. So how does fight or flight come in? Well, first you'd have a trigger from your environment, and generally it would be something that scares you or something that you need to do something about. You either need to, certainly in evolutionary times, fight it or run away from it. So you have a trigger that is picked up by your hypothalamus, and your hypothalamus then triggers your sympathetic nervous system. So this is the one that gets you ready to go. So adrenaline, which is a stress hormone, is released from the adrenal medulla which is in the adrenal gland, when your hypothalamus picks up your trigger. So there's a link here between the endocrine system and the autonomic nervous system. That obviously increases your heart rate because your sympathetic nervous system kicks in, makes the body ready to fight or flight, makes it ready to punch it or run away from it, whatever that may be. And that's a brief overview of the system. Obviously, it kind of makes sense from an evolutionary point of view that would happen if there was a predator in the environment. That would make sense. But actually, modern day things, it still has an effect. You get the butterflies in your stomach when you're nervous about your upcoming psychology exam. Good luck, by the way. Or your public speaking, or whatever it is that you're worried about, and you can't punch it or run away from it. I wouldn't suggest running away from your exams. So it's maybe a bit redundant. So that's maybe a bit of evaluation. Next then, we've got more information about neurons, brain cells, which some of you have more of than others. generic statement there. So neurons allow signals to be sent generally between the peripheral nervous system and the central nervous system. So basically between your senses, your skin receptors, and the brain. So there are different types of neurons. So you've got sensory, relay, and motor, and it generally fires in that order because your sensory neurons pick up messages and carry them from the peripheral nervous system to the sensory nervous system via receptors. You've then got relay neurons, which connect sensory and motor neurons. So it relays and other relay neurons, and it transfers the information. So it's like a connector, interneuronal messages get sent. You've then got the motor neurons, and they are the ones that send the message to the glands and the muscles to have an effect, so moving your muscles or releasing the glands. So what is the structure of a neuron? You also need to know that. So first you've got a cell body. So this is the main part of a neuron. It's the big bit with a circle in the middle on the diagram on the right hand side. So that's there's a nucleus in there which contains all the genetic material of the cell. You've then got the dendrites, which are the branches, and these carry nerve impulses from neighbouring cells to the cell body. So they're the excuse me, they're the branches that are coming out from the cell body that you can see on the right there. You've then got the axon, and that's the long, thin part of the cell, and that carries information away from the cell body. So it's taking the message from one neuron, taking it down the cell body, and sending it to the other. The axon is covered in something called a myelin sheath. Think of it as kind of the plastic covering on a metal wire. So it allows messages to be sent better, it conducts it, it protects the neuron as well. It's not in one block. It is split into several different blocks to allow the information to be sent better, and then it finally goes down into the axon terminals, the terminal buttons. So that's the end of the axon where it will send information via the synapse, which is the gap between the axon and the dendrite, to the next cell, and we'll talk a little bit more about the synapse in a moment. Within the cell, the information that's transferred is via electrical transmission, and there are ways of measuring that, and again a bit later on we'll talk about brain imaging techniques, some of which pick up the electrical transmissions. There's a little bit of information that you need to know about neurons being negatively charged, and when a neuron fires it becomes more positively charged, and the changing from negative charge to the positive charge is called action potential. So we'll talk a bit more about that now. So when the information has been received by the cell and has travelled electrically down the cell, what you get is synaptic transmission. And this generally happens via a chemical process. So within the cell it's electrical, but between cells it's generally a chemical process. And these chemicals are known as neurotransmitters. Again, neuron, brain, and transmitters, well, you transmit information, don't you? So it's a bit like that. So what will happen is the information travels down the axon to the axon terminus to the synapse, and then the information is transferred via the synapse or synaptic cleft from the axon of the sending neuron, known as the presynaptic neuron, to the dendrite of the receiving neuron. That's known as the postsynaptic neuron. Neurotransmitters are contained within vesicles, which are almost transporters with the chemicals inside, and these vesicles are released from the presynaptic neuron. So in the gap between the two neurons, very, very, very tiny gap. And what will happen is then on the receiving neuron there are receptor sites, and there are different receptor sites depending on which neurotransmitter is being sent. So the receptor would only receive information from particular neurotransmitters. Lots of different neurotransmitters, you will have come across them in the rest of the psychology course: serotonin, dopamine, etc. So the specific vesicles and receptors are different. There's two different types of synaptic transmission. There's excitation and inhibition. Excitation make neurons more likely to fire. They're positively charged, and neurotransmitters such as adrenaline are excitatory. You then have inhibitory transmission, and so this makes it less likely to fire, so they're negatively charged, and serotonin tends to have inhibition. Next, we go on to the year 13 content. So what we've done up until now was the year 12 content. You didn't need a great deal of evaluation. Evaluation then becomes a bit more important now. So first we start with localization of function in the brain. And so there is still a question suggesting, are specific areas of the brain responsible for certain functions? And that's still a bit of a question, and there's still a bit of debate over that. So we'll look at kind of the for and against. So in general, people seem to think that the hemispheres, so the left and right hand side of the brain, some are dedicated potentially to some actions rather than others. So this is called lateralization of the brain. And so generally what you find is actions in the left of the brain control the right side of the body, and the right of the brain controls the left side of the body. You've also then got your cerebral cortex. So that's the outer layer of the brain, sometimes called the T-cozy. It's about 3 millimeters thick. Known as your grey matter, and it's what's thought to separate humans from other animals. It's what's thought to give us a higher cognitive thinking and processing power, higher functioning, and it's thought to develop later. So the basal areas of the brain at the bottom of the spine, at the top of the spine, sorry, they developed first. They develop our breathing and control. It's the cerebral cortex that has our higher functioning and processing and cognitive abilities. You've then got the lobes, and this is where you've got the different areas of the brain. So you've got the frontal lobe, which is represented by the red area in the diagram on the top right hand side, and that's thought to be involved in motor. It's the motor area, so involved in movement. And so obviously if you have injury to this area of the brain, then you'd have difficulties with movement. You've then got your parietal lobe, and this is the yellow section on the diagram on the top right hand corner. This is thought to be the somatosensory area, and so this is detect your senses, so from the skin, so your heat, your pressure, etc. And if you have difficulties in this area, then obviously you'll have difficulties with that side of things. But actually what you find here is that the more that you use the area, so your hands for example and your face, the bigger the somatosensory area is, and so we have a bigger area to account for more information coming in from our face and our hands. You've then got the occipital lobe, and that is the green area on the right hand side, and that's our visual cortex. And so what we have here is that the visual field, so your right visual field goes to the left side of your brain and vice versa. And then finally we've got our temporal lobe, and that's obviously the blue area, and this is our auditory centre, and so again damage to here would affect our speech and our ability to detect words and language. What you then have is, we know a bit more detail about the language areas of the brain, which are thought to be on the left hemisphere. So you've got Broca's area, which is in the frontal lobe, and this is responsible for speech production. And what you have is a disorder known as Broca's aphasia, and so that's if you have damage to this area. And what that will produce is slow and laboured speech. People won't make much sense. They'll be speaking, they'll be trying to get something across, but you won't understand what they're saying. And there's a case study known as TAN, and it was a man who could only say the word TAN because he had Broca's aphasia. And then you've got Wernicke's area, and that's in the left temporal lobe, not load, God, my spelling is atrocious. And this is involved in language comprehension. And then you get Wernicke's aphasia, and so this is where people have difficulties producing language, because of the language comprehension difficulties. So they can still produce language, but they might not understand it. Did I just contradict myself there? I think I did. So sorry, this is where people have issues producing language, but they can't understand it. So they'll say nonsense words. They may sound fluent, but actually they're meaningless. So what evidence do we have for any of this localization? Are different areas of the brain associated with different things? Well, there is certainly some evidence. We've got brain scans. Peterson et al. Conducted brain scans and asked people to listen to things and asked people to read things, and what you found is different activation in Wernicke's and Broca's area, which suggests these areas are different, and you have got this localization of function, specific areas responsible for specific actions. You've also got case studies, such as the case of Phineas Gage, who had the railrod go through his head, poor bloke, and that ended up taking out his left frontal lobe. And what happened is that he changed, his personality changed massively. Friends and family said he's no longer Gage, he's no longer what he was. He went from being quite a nice, mild-mannered man to being quite angry, and so it had effect on his lobe, and again, so it had an effect on his mood. His left frontal lobe had an effect on his mood. So this supports localization of function. You've also got Doherty et al. They tested 44 OCD patients, and they were given a cingulotomy, which is where your cingulate nucleus was cingulate gyrus was there was surgery on it, and so that still happens. There is still surgery. It's quite controversial, neurosurgery, but it tends to be in the most severe cases when other treatments haven't worked for OCD. And actually, that seemed to work in these patients. They seem to respond to this, and so there's practical applications here of knowing this localization of function. We can potentially help. It's not all plain sailing though, because Lashley conducted studies on rats and actually found it didn't really matter which areas of the brain of rats that he conducted surgery on. Actually, it seemed they seem to think that there's a more holistic view, certainly in rat brains, and so that goes against the localization of function. And also, we're about to look into a bit more detail about plasticity, and this is where some areas of the brain take over the role of other areas. Well, that goes against the idea of localization. It's not so local if any area can do any action. So that leads us quite nicely into plasticity and functional recovery. So what we mean by plasticity, sometimes called neuroplasticity, is, as I just said, it's where changes in the brain or the brain changes and adapts to different areas, and this is generally after trauma or injury. And so what happens is the areas that we use tend to strengthen, and you get stronger connections and stronger bonds there, and the areas not used tend to die off. That's known as synaptic pruning, and it's thought that this happens throughout life. There are times where it happens a lot more during childhood, but actually it's thought to happen throughout life. Lots of studies to support this. The most famous one is Maguire et al.'s London Taxi Driver Real study, and what they found was that London taxi drivers, they have to pass something called a knowledge, where they know all the routes in London, and what you find is their hippocampus, which is an area associated with memory, grows. So the brain actually changes. It's fascinating, amazing. Psychology is awesome. The brain changes based on the environment and the stimulus that you're getting. Draginski et al. Also found changes. So this was medical students, took their exams, and what you found was changes to their posterior hippocampus and the parietal cortex. The brain changes based on there's plasticity happening based on what's happening in the environment. And again, that was found by Medchlowny et al. Larger parietal cortex in bilingual individuals, again an area associated with language and learning languages. You also get functional recovery, and this is, as I said, more linked to trauma. So function is transferred from damaged areas, which are injured by knocks or strokes or something, to other areas. And what you find is sometimes this happens straight away, and the brain adapts really quickly. Generally, that's when you're a bit younger. Or you can have rehabilitation and kind of occupational therapy and be almost trained for that to happen, which is good. The way that happens, it's thought to occur by creating new synaptic connections, known as axonal sprouting, reforming blood vessels, and recruiting homologous areas, so areas that are associated but on the opposite hemisphere. So does this happen? The evaluation. So obviously there's great application here. Neurorehabilitation and therapy can really help to stimulate areas and growth and functional recovery. You've also got an issue though, in that you've got negative plasticity. So all the things we spoke about there are quite positive: recovering from injury, your brain responding to learning the knowledge or learning medical exams. Well, actually it can happen the other way as well. So if negative things are happening, so drug use for example, and you find the brain adapts to that, and that's not what you want. You've also got something called phantom limb syndrome. So if people have lose a limb or have amputations, they can often feel that their limbs are still there, and it can be quite painful and obviously mentally scarring as well. And that is the brain reacting, so the brain changing. Plasticity, not always a great thing. Age is a big factor, so it is thoughted that it is thoughted. God, issues. It is thought that age, when you're younger, plasticity happens more and functional recovery is more likely, but it is thought to occur into adulthood. And Brazola et al. Found this. They looked at 40 to 6-year-olds playing golf, and fMRIs showed reduced motor cortex activity. Which suggests that they're getting better at it. They're not needing to use their motor cortex because actually they've learned how to play golf. There's also something called cognitive reserve. So this suggests that the more educated you are, the better. So the stronger your connections are in the first place, the better your recovery will be afterwards. So Schneider et al. Looked at the difference between people who have 16 years worth of education compared to 12 or less, and found about 40% recovery in the better educated people. So there you go. Stay in education, kids. It can help your brain recovery later in life. Okay, so next we then talk about split brain research into hemispheric lateralization. This is essentially the research of Sperry. Hemispheric lateralization is the idea that some brain function is controlled by one side of the brain rather than the other. So Sperry looked at patients who had their corpus callosum cut. So that's the bit down the middle of the hemispheres of the brain. Really, really unfortunate for them. It was due to epilepsy, but really handy for research, because if the brain, if the corpus callosum is cut, then the two hemispheres can't interact with each other. So it tells us a lot about which side of the brain does what. So what they found in these patients was that they could describe objects in their right visual field. Don't forget that the left-hand side of the brain controls the right-hand side of the body. So the right visual field leads to the left hemisphere. So they could describe objects in the right visual field, so the left hemisphere could describe objects, but not in the left visual field. So the right hemisphere wasn't able to describe the objects. So this supports the idea of lateralization of language. When they asked to them to do a touch task, the object could be selected, so this seemed to be unaffected. When they were presented with different words, key and ring for example, so key was presented on the left visual field, they could write key with the left hand. So this looks like the left hand, again left side of the body, is controlled by the right hemisphere, and again suggests that there is this distinction between the left and right hand side of the body and the brain. For faces, it seems as though the right hemisphere is in control, as they selected faces from the left visual field. So what does this all tell us? Well, research report in general, the left visual field is more analytical in general, and the right visual field, the left hemisphere is more analytical, and the right hemisphere is more to do with spatial and musical tasks. The research methods used tend to be quite good. They were reliable, they were standardized, you could repeat them with those participants, and they also tend to be quite valid. There was a high level of control, internal validity. There is still debate, however. Obviously it was a small sample size. Is there generalizability here for, these were patients that had their corpus callosum cut? Does that apply to everyone? And actually some still argue that there is duality, which is that there are two minds, whereas others argue that actually most tasks use both hemispheres of the brain. Next, we move on to ways of investigating the brain, and there are 4 that you really need to understand. So what we're talking about here is, well, what's going on in the brain, and how do we test them? So fMRI, functional magnetic resonance imaging, measures the blood oxygenation, and can look at that in the brain when you're doing a particular activity. So if there's more oxygenated blood in a particular area, it suggests activity is happening in that area. So you could give people different tasks to do, whether that's reading, or whether that's singing, or whether that's visual tasks, and whatever area is active shown by the fMRI, that suggests that that area is associated with that function. It can produce 3D maps or 2D maps, showing what is happening, and so it does show this localization of function, as we've been talking about already. The strengths of fMRI there, it's relatively risk-free, it's non-invasive, you don't have to have any surgery or anything to have it done. You can create these great high resolution, really clear images, which is fantastic. However, you do need an fMRI machine, which tend to be quite expensive, and also it's quite sensitive in the fact that patients have to stay still, and if patients don't, it reduces the quality of the picture. There's poor temporal resolution, and what that means is to do with timing. So there's a delay, a lag between what's happening and the brain's reaction. So sometimes that makes it difficult to see actually what area of the brain is affected when. And what it tells us is general regions rather than specific neurons or neural connections, so it's not as detailed as it could be. So it's great, but there are some images. Next, you've got electroencephalograms, so EEGs. Here you put a skull cap on. The skull cap has lots of little sensors that attach to the skull. I've had one actually, there's quite a strange sensation, little gel on them. And the electrodes measure electrical activity, and what it produces is patterns and waves, and you can see electrical activity from the neurons happening in the brain. So what they can show is abnormalities in wave patterns, so you can have control groups where you see what is happening, and you can then compare that to different groups, maybe say that have certain disorders or epilepsy, and you can see the differences in the brain, so it could detect whether something's going wrong in the brain, comparing individuals to healthy controls. So this is really good for measuring things that are to do with brain waves, such as epilepsy and sleep, and this one has much better temporal resolution than fMRI. It's happening as it's happened. There's a milliseconds lag, sometimes less, and so it's really good. It's really in the moment. However, these waves are quite general, and you can't see specific areas, and that is a bit of an issue. However, you can get over that by using event-related potentials, ERPs. So this is the same as an EEG. You've got the skull cap on, but here individuals are presented with specific stimuli. So a task to do, something to look at, for example. And what this means is that the waves can be more specific, so you can average out responses from what people are doing when particular stimuli are presented. And so we can know which brainwaves are triggered by particular stimuli. That's really interesting. So it makes EEGs more precise, which is a strength, and it still has that good temporal resolution, which is brilliant. But again, it's not perfect. There are different methodologies used by different people, different researchers, different EEG machines lead to different outcomes, and sometimes it's hard to control stimulus presentation. What I mean by that is that there could be background noise. It's hard to have full control over exactly what's happening. So are we sure that those brainwaves represent just the stimulus that's being presented? Very, very, very difficult. And last but not least, for a reason, we've got post-mortem. So this is where, after death, an individual's brain can be looked at, can be dissected, removed, and you can look at what's happening and compare them to healthy brains. So HM was a great example. Henry Molaison, from the memory topic, his brain was cut into lots of different pieces after he passed, and told us a lot more about what was going on and his memory issues. It tends to be when people have specific injuries that you might want to look at their brain afterwards. So this was one of the first methods, and so it led to some of the others, so that's a positive of it, and it improved our specificity and improved our understanding of the brain to begin with. However, causation is difficult. We don't know whether certain abnormalities are causing certain behaviours that we knew the participant was displaying when they were alive, or are they as a result of changes in the brain. And there's also an issue of consent, and I obviously don't mean asking a dead person, can we take your brain out? What I mean is that, is there consent beforehand? And even if there is, you're talking about people here with brain injuries. So do they know enough of what's going on to actually give their consent? So that's a difficult one to know. Okay, and then we get to the last part of the biopsychology course. So that's circadian, infradian, and ultradian rhythms, and endogenous pacemakers and exogenous zeitgebers. Easy for me to say. So first we have circadian rhythms, and so these are rhythms that happen once every 24 hours. And so the most likely one that we've got is sleep. You generally sleep once every 24 hours, sleep-wake cycle. So you've got your internal body clocks, which are your endogenous pacemakers, and then you've got the external changes, which are the exogenous zeitgebers, and these are thought to control our circadian rhythm, certainly our sleep-wake cycle. So obviously we tend to be more drowsy at night and more alert during the day, and light is thought to play a part in this, in both the endogenous pacemaker and exogenous zeitgeber. We'll come on to that in a bit later. Sif was the caveman who, not like Barney Rubble style, but a man who spent time in a cave, spent 2 months in a cave with no light to see the effect it would have on him. So he's only got his body clock, his endogenous pacemakers to go by. He's not relying on the light. And what was found that he generally kept a 25-hour cycle. So that's a bit longer than our normal day that we might obviously expect to find with the exogenous zeitgebers, but what was found is he kept a regular sleep cycle. So this suggests that our endogenous pacemakers have maybe more of an influence on us. Ashcroft and Weaver had participants spend 4 weeks in a World War II bunker with no light. Sounds like a spin-off of Love Island. But what they found here was these participants showed a sleep-wake cycle of around 24 to 25 hours. So this suggests our exogenous zeitgebers do have a role to play, because they kept the same sleep-wake cycle as you would expect when you had the external cues, but they didn't have the external cues. Folkard et al., similar sort of study. This time they put 12 participants in a cave for 3 weeks, no light. They did have a clock, and they were told to go to bed at 11:45 and wake up at 7:45. So the clock would be an exogenous zeitgeber. It's a cue. They didn't have the light to go on. And what they found was what the researchers actually did, they changed the times unbeknown to the participants, and changed it from a 24-hour day to a 22-hour day. Lots of the participants really struggled with this and couldn't adjust. So this actually suggests again that the endogenous pacemakers are really, really important when it comes to circadian rhythm, certainly, because it was hard to overpower the exogenous zeitgeber of the clock. So what does this all mean? What's the evaluation? Well, there's application here to shift work. It suggests that we have a body clock. We kind of need to stick to it. So if you're working nights, then that's a bit of an issue. And what you find is, regardless of whether you normally work in the day or at night, there tends to be this concentration drop around 6 a.m. It's known as a circadian trough. And also sleeping nights links to poor health. So there's maybe a link to your research methods. I dare to say that word. But to the economy. So what does it mean for night workers? What does it mean for companies that need people to work through the night? And actually maybe a bit of health and safety as well, what's happening at 6 a.m. There's also interest in drug treatment here. So when do we have our drug doses? So we have circadian rhythms that link to our heart rate, our digestion, and our hormones. And if we're taking drugs that affect those things, then we need to be aware of that. And pharmacokinestetics have borrowed this information, and they've given suggestions for timings and doses of drugs based on what we know about circadian rhythms, our natural body clock. And then did a bit of evaluation for the case studies. They obviously tell us a bit. It's good to know this information, and sometimes actually case studies tell us a lot, but actually can we generalize these results to wider populations? Quite difficult. Were there individual differences in these groups? Potentially. Okay, next we move to infradian rhythms, which happen less than once per 24 hours, and we'll look at monthly and seasonal cycles, and then ultradian rhythms, which happen more often than once a day. So, infradian rhythms, the two that you need to be aware of, one is the menstrual cycle. So these are monthly changes in estrogen, which is a hormone that leads to ovulation. This happens once about every 28 days and is an endogenous pacemaker. Stern McClintock found pheromones led to what we call menstrual synchronization. So this is where females would menstruate at the same time. The way that they did this, it almost makes you want to vomit, so I'm not going to go into a great deal of detail, but it involves putting pheromones on the lips that have been under armpits and all sorts. But anyway, it supports it. The next one that you need to be aware of is seasonal affective disorder, otherwise known as SAD. Quite ironic, considering what it's describing. And this is where people find people who have SAD disorder have low mood in the winter. It's known as a depressive disorder, and it's a circadian rhythm, so obviously it happens once a year because it will happen during the winter months, and it's linked to melatonin secretion, which influences serotonin. And again, it's an endogenous pacemaker. The body reacts to the light, to the season, which affects the melatonin, which again affects the serotonin. Then we've got the ultradian rhythms, which are the stages of sleep. So we sleep once per day, so that was a circadian rhythm, but within our sleep there are 5 different stages, and these happen over 90 minutes. So you might sleep for 6, 7, 8 hours, hopefully lots when you're revising. So within that time, you go through several ultradian rhythms of sleep, and there are different stages, which are each related to different brainwaves, which we can see in an EEG. So first, you've got stages 1 and 2, and these are light sleep and the beginnings of sleep, where you see what we call alpha and theta waves, and these are different waves that have different patterns, and so we can see these are different stages of sleep. Next, you've got stages 3 and 4 of sleep, which is the deep sleep stage, which again is another type of wave known as delta wave. These are slow waves. You see bigger gaps between the highs and the lows. And then the final stage, the 5th stage, is REM sleep, rapid eye movement. And this is the deep sleep. This is where your body's paralyzed. You might see rapid eye movement, funnily enough, quick brain activity, and it's actually the stage of sleep that is most associated with dreaming. So, evaluation, do these things happen? Well, there is some support. An evolutionary theory might suggest that there is an evolutionary basis for menstrual synchrony. So it would suggest maybe that it's an evolutionary advantage for females to synchronize their monthly cycles, because that would then link to pregnancy, and maybe they would be pregnant at the same time, and so they could all help each other to be mums. Well, actually, if you think about that in more detail, it actually almost goes against evolution, because what you would have there is more competition for mates. So actually, from an evolutionary point of view, it would make more sense for females to ovulate at different times. So is that actually a valid explanation? And could there also, in the Stern study, be confounding variables? Are there other factors other than the pheromones that might have influenced why the females in that study synchronized? So it's not cut and dry. There's also support from animal studies. Other animals have been seen to have these cycles and release of pheromones at different times, and sea urchins are one of those. There's also a link to the different brainwaves in the different stages of sleep. So EEG, Dermot and Kleitman looked at EEG patterns at the different stages of sleep, and they did find different brain patterns, and this supports the idea that there are these different rhythms, different stages, and again this happens in 90 minutes, and so we do have these more than once per 24-hour rhythms. Finally, we've got obviously the seasonal affective disorder. Well, if we think that part of the reason is our exogenous Zeitgeber in light, well, could we maybe increase the light? Could we give UV or phototherapy to try and help people who have seasonal affective disorder? And it's been found to be very successful, and suggests that there is these bodily reactions that can be affected, and obviously we can then get treatment for. Last and not least, then, we've got endogenous pacemakers and exogenous Zeitgebers. So, endogenous pacemakers. So these are body clocks, essentially. We've got the suprachiasmatic nucleus, which controls the sleep-wake cycle in the hypothalamus. It's thought to detect light even when our eyes are closed, so it can do it when we're asleep. wake us up, and it adjusts the body clock accordingly. Bit of research by some horrible people. De Courcy et al. Destroyed poor little chipmunks' SCN, which interrupted their sleep-wake cycle, and then they all got attacked by predators and killed because it's thought that they were awake when they shouldn't have been. So that supports the idea that the SCN is really important. You've then got the pineal gland and melatonin, which are linked to seasonal affective disorder. So these are the body's clocks, the body's internal processes that have an effect on our biorhythms. You've then got exogenous zeitgebers, which are external factors leading to entrainment. So light is the obvious one. It influences the SCN. Campbell and Murphy found this. They also found that skin receptors had an effect on sleep-wake cycles. So if you shine a light on the back of someone's knee, the skin can pick up light signals, and that can have an effect on sleep-wake cycle. You've also got social cues, so meal times and bedtimes can have an influence on how awake people feel. So influence infants certainly, but also when we're jet lagged, if we fly to another country, when we eat and things like that, has an effect on our body. So what's the evaluation here? Is the SCN as important as we would suggest? Well, no, maybe it's not as much of a master clock as we thought. There are other things, skin, lungs, liver, etc., that also have a role to play. This was found in a study on mice as their biorhythms were changed, cells in the liver leaving the rhythm of the CNS unaffected. So you can change sleep-wake cycle with the CNS being intact, but other areas are affected. There was obviously bad ethical considerations in mutilating chipmunks, and can we actually apply the results of those studies to humans? Extrapolation is an issue there. Are exogenous zeitgebers overstated? Potentially, social cues don't always lead or don't always alter the sleep-wake cycle. So people who are blind, who obviously can't pick up those cues, they seem to be unaffected. People who live in the Arctic, so where light is affected at different hours of the day, they also seem unaffected. So maybe the endogenous pacemakers have more of a role to play. Realistically, it's probably an interactionist approach where actually in the real world both things have an influence and both have a role to play. Thank you very much for listening, and best of luck in the exams. You'll be fine.